Data in the world is growing at this exponential rate. I Googled a few fast facts of how much data is being generated daily, and I thought a few of these stood out quite prominently. In 2021, there was an estimated about 2 trillion Google searches by the end of the year. On WordPress, we're publishing around 70 million unique blog posts, and with that comes around 77 million new comments. This was in 2021. And in 2020, it was reported that people sent 500 million tweets daily. And I just want to emphasize the amount of data that we get from all of this being generated on a daily, monthly, or yearly basis. Now, the advent of all this data has resulted in generative models or artificial intelligence that can learn from this data. So a really wonderful example of this is Bing Chat. This is an AI-powered copilot that takes in a lot of these data sources and provides us with a copilot or a helper that can answer a variety of different questions. So for example, this is an image I generated using Bing Chat with this simple prompt, asking it to help me generate an image that visualizes the amount of data that's being generated in the world. Now, we can imagine a scenario where we can apply these same concepts and use the same idea that we're generating a ton of data in order to revolutionize biology. So this is a made-up scenario, it's not real, but you can imagine an example where we ask an AI assistant to generate 100 new designs for protein sequences that can catalyze specific reactions. Our AI would then provide us with these predictions for protein sequences, which we could then test in a lab. This idea sounds a little bit crazy, but is not so far-fetched. Francis Arnold in 2018 won the Nobel Prize in Chemistry for this concept of directed evolution, where you can identify a parent enzyme that kind of does a reaction that you might be interested in. In this case, they used this is an example of P450. This is an iron-containing enzymatic catalyst. And then with some user intervention or direction from the scientist, we can conduct these successive mutation cycles, which are denoted by these colored regions here, using some informed prior about how they impact the catalysis of the reaction that we're interested in. And we can iterate over this again and again until we achieve some desired function. And in this example, they were able to evolve a new enzyme that was selective towards a particular reaction, and it was environmentally friendly. And this had not existed prior to this process. So when we think of using AI to intervene in processes like this, we think of two major classes of deep learning. First, we have these discriminative models that can discriminate between different kinds of data instances. So for example, this is like a common example of a sample of a bunch of chihuahuas, but also these blueberry muffins. And the goal of your model in this case would be to discriminate between which of these are photos of dogs and which of these are muffins. As humans, we have a general understanding of what it is that makes something a dog and not a muffin, but machines are not informed with this. So with generative models, the aim is to generate new data instances by learning these latent representations of what it means to represent that data. And in this case, intuitively, we can link this concept of a latent space, which is this kind of abstract concept, for example, of what we consider a dog. And the goal of our generative model is to encode a meaningful representation of this. Now, generative models do this by compressing our data into a low dimensional space before reconstructing them. And that thing that it's learning in the process of this is what we call our latent space. Now, there's a few ways of doing this. Conventionally, we've used these things called GANs or VAEs, which generate images in a one-shot fashion, meaning they're given a task and in one swoop are trying to predict what it is that makes a dog a dog. However, this new class of models that's kind of gaining traction are these diffusion models, where instead of doing this task in a one-shot fashion, we're now generating images or learning these latent representations through iterative refinements. Now, today, this will be the focus of this talk of, what are diffusion models, but just to give you some, prior of like why we're interested in doing diffusion, it's because these GANs and VAEs suffer from a few critical problems. First, they're quite difficult to train. They're prone to overfitting, and they have poor generalizations. We find that they can't work that well outside of their given tasks. However, on the other hand, when we reformulate our generative models in this diffusion framework, we find that we get high quality generations, they're diverse in samples, and also we're able to train these a lot more stable. So it requires fewer like engineering tricks to get them working. Okay, so with this overview, let's dive into what is a diffusion model exactly. A diffusion model is composed of this forward and reverse process. So here I'm describing the forward diffusion process. It's the process by which we take input data that's represented as this x naught variable, and over some fixed series of time steps, we're going to convert this to something that is easily sampled from, say, a Gaussian distribution. The reverse process then generates data by denoising the samples at time t, up until we get new samples at time 0. So this is just to like orient you with directions. The reverse process is often denoted by this distribution labeled p, whereas the forward process is denoted by this variable q. Now, the concept behind this is that we're taking something that can be represented by a complex distribution, and we're now converting it to something that we can sample from a very simple distribution. And to depict this or give you some kind of visual understanding of what's happening, we can use a test case of images. So taking this image of a cat, let's say we pulled it from our data set, we're iteratively adding noise to this image up until this time xt, where this sample is indistinguishable from our beginning product, and more importantly, is easily sampled from a standard Gaussian. Now, for the reverse process, the goal of this is to now take this thing, this image that we can easily sample, and undo the noise being added at each step to then reconstruct that input image. This is our reverse process. However, there's no easy way to kind of reverse these transitions over an entire data set. So this is where our neural networks or our AI solutions come in. Now, I'm going to walk you through some math-heavy slides, but the takeaway is that in order to learn this reverse process, we need to resolve 3 key components. First is we need this scheduled way of noising the inputs from x naught to xt, meaning we need to be able to define a series of time steps upon which we can add noise in a way that's controllable. The second thing we want to accomplish is defining a tractable way of sampling an image at any time step, given just our input image. And then the last thing we want to do is define our elbow, or this is our evidence lower bound. It's going to be our function that we need to perform training of our neural network. Okay, so for this first task, we're defining this fixed noising pattern. And to do this, we introduce this variable called a variance schedule. It's defined by this term beta tau. And this is the rate at which we're adding noise from step t-1 to t. This can be represented by a function as simple as a linear example here. So this is a linear rate of noise addition over a series of 1,000 time steps. Okay, so now that we have a controlled way of adding noise, we can then define this function here. This is our probability of sampling something at time t, given the sample at t-1. And now we're using this variance schedule parameter to control the mean and the variance of that distribution. So this is our controlled way of adding noise into our system. Great, okay, we've accomplished our first task. We now have this scheduled way of iterating from x naught to xt, where no matter what we choose as variable t, we can control the noise being added. A kind of side point here is that the way that we add noise into these systems can greatly impact how well that they learn from their training task, but that's kind of a whole field of research that's separate from this. Now, the problem with this is with this definition, it requires that from our input sample, we then have to sample every single time step to get a sample at xt. But during training, this can be quite tedious. We don't want to have to sample every time step in order to sample various images in this process. So to do this, we're going to rewrite this function as a way of approximating a sample at any time t, given just the input sequence. To do this, we're applying this thing called a reparameterization trick. This is going to enable tractable closed form sampling at any time step. For the sake of this talk, I won't get too deep into the weeds here, but I highly recommend this course, Intro to Deep Learning. It's run by Ava Amini, who's presenting later, and Alexander Amini. And it's like quite a nice resource in learning these concepts from the ground up. But in essence, what this is doing is making our model much simpler to train. Now I'm going to introduce some new terms here, but really what I'm just doing is re-representing our variance schedule that we've defined earlier in a couple simple terms so that we can like easily access them. And so here we're just doing these math tricks to help us rewrite this noising schedule in terms of things that are a little bit easier to calculate. But the takeaway here is now this alpha t term just represents our noising schedule differently. And now we've introduced this cumulative product of all the scheduling values at that given time t. And this is just the cumulative product of all of those at each time step. Okay, and so this is where things get a little crazy. We apply this reparameterization trick that we introduced earlier, which is a bunch of math, which if you really want to know, Lillian Wang has this nice depiction of how to derive this, and that's linked below here. But what we end up with at the end of this is a way of sampling our image x of t, given just the input itself. And this is due to taking the product of that noising schedule and removing this eta factor, which now represents our noise. This is what we've done in our reparameterization trick. What this does is it enables efficient sampling during training, meaning that, again, we can sample any image at any time step while we're training our neural network. And it's now given us a tractable reverse conditional probability, which makes the training of the neural network quite a bit easier. Okay, so this idea of a tractable conditional reverse probability is important because now we can much more simply compute our gradients and run backpropagation. So again, this derivation here is really nicely depicted in this blog post here by Lillian Wang. But just know that this has a lot of similar themes of how we solve maybe a VAE. And so the math behind this is not necessarily new. This first part here is just using the idea of a variational lower bound to optimize our negative log likelihood, which we can write as this. And then the next part of this comes from a paper in 2015 by Sol-Dickstein, where they evaluate this to be analytically computable for a diffusion process. And that's how we end up with this equation here, which looks quite complicated, but actually makes training our model quite simple. So let's quickly just break this down a little bit. This term, the loss of the lower variational bound, is composed of 3 terms. I spilled. The first is just defining how close a sample at x of our final time t is to a standard Gaussian. So this is taking a KL divergence, which is a measurement between 2 probability distributions, and saying that, okay, at time t equals big T, is this a standard normal distribution? What's cool about this is that because Q doesn't have any learnable parameters in this case, as long as we're accomplishing this objective, we can ignore this during training. The second term here is just a reconstruction term. It's similar to one that we define in VAEs, and we have known ways of treating this in our models. And then this part of the equation here is kind of the bread and butter of our diffusion model. It's the difference between the learned denoising steps, so things that our model is predicting, and what we approximate to be true about the reverse denoising steps given by Q here. Now, what happens is in this paper from Ho et al. In 2020, they find that we can train models by deriving a simple objective, here denoted LT simple, that just works given this following equation. And what they do in this paper is that they reparameterize this term here in order to just be predicting the noise being added at each time step t, given your inputs. And this is like quite a neat trick. So in reality, what our model is going to be doing, given this function here, is learning the applied noise at each time step. Okay, and this is great. I've highlighted it in pink here because we're going to reference this in a little bit. But the key idea here is we now have this loss function to train on. Okay, so what does this mean in practice? How does this look like when we actually want to train a neural network? Well, we'll just go through the steps here. It's actually quite simple. We pre-compute those noise scheduling terms that here we're calling alpha t and bar alpha t. We sample some time step t for our data, and then we generate a sample x of t, so a noised image in this case, given the fact that we know the original image and we've predefined this noising schedule. We can then train our model by calculating the gradients for our loss function, and those come from the idea that we've now defined this loss function L simple that we just kind of went through the derivation of. And again, our model is predicting this term E theta, which is the noise being applied at this time step. Okay, and now we just iterate and repeat this until our model converges. So we measure kind of our loss over time and use that to decide when our model is done. Okay, so now we've trained our model, and the question is, how do we generate new data points? So now we can start with some random sample sampled from our Gaussian at time x big T. We can iteratively run it through our neural networks to denoise the inputs from x of t minus 1 to x of, oh, that should say x of t to x of t minus 1. So we're denoising in the reverse process. But if our model's predicting noise, how do we get this prior term given the current term? And the answer is just more math. So this is another derivation that we find in Ho et al., where they define a way of just generating the sample through denoising. And this is the reverse process here. Okay, now that we have this, we iterate in reverse up until we reach time equals 0. And ideally, we now have new samples of a cat on this data set trained on cats. Okay, so that's great and all, but often what we want is to control the output of our generative models. So if I train a model on images, often I want to control what the output of that is. And so in order to do this, we need to retrain a model to denoise data given these data conditioning pairs. And here's some popular examples that arise here. The top is this text to image generation with a software called Imagen, which is trained on these pairs of images from a data set. And in particular, these are boundary labeled images. So they have quite high context about like what the things in the image are. And then at inference time, we can then provide that conditioning tag and ask it to generate something totally new. So in this example, we ask it to generate a cute corgi that lives in a house made of sushi. This is obviously not something that our training data would have seen, or we would not have expected it to see. And we can show a bunch of these crazy prompts and see that our models learned quite well. Now we think about like, how do we control these generations? And the answer lies in providing these conditioning pairs where you have a label associated with an image. And we can do this with image to image as well. So we've seen this example of text to image where I can generate an image given a conditioning tag, but then I can also do different tasks like provide a grayscale image, train on that with the conditioning tag of its original coloring, and then use that at inference time to then color what the model thinks that this image should be colorized as. And this is an example from 2022 called Palette. Okay, so we've described this example on image data of how we conduct diffusion, but a more relevant example, and one that Ava will dive into later, is this idea of diffusion in discrete spaces. So in cases where your data is composed of k categories of entries, this could be, for example, like a binary bit problem in chess, where you would want to move on an 8x8 board and not make continuous moves on this board. and then a relevant example being protein sequences. We can think of protein sequence design as this discrete problem because functional diversity in protein sequences arises from this K to the L combinatorial space, where K are these categories represented by the 20 natural amino acids that exist in a protein sequence, and L is just describing the protein sequence length. So similar to our continuous model, the formulation of this is the same, where we're taking an input and slowly corrupting it out, but the implementation of this now looks slightly different. So here we have a natural protein sequence that we're mutating at each time step until we get a sequence that's essentially sampled from a uniform distribution, meaning that we can sample every amino acid with likely probabilities. And again, in this case, we're defining these three criteria that we need to then train the reverse process. And these are the exact same as the continuous case. We're just defining them now for the discrete models. So starting here, we want to define a forward process with a fixed noising pattern. And for those who I think struggle with the continuous case, the discrete case is a little bit more intuitive because some of this comes from, all of this comes from Markov math, but it's quite easily represented here. So to sample a sequence at time t, given the prior time step, we can take that sample. I think this is labeled wrong, but we'll take the sample at t-1, multiply it by some Markov transition matrix, and then get the sample at the next time step. And we can get this by taking a categorical distribution over that probability. Again, in this case, we're choosing some matrix M now instead of alpha that's defined in such a way that we can control the noise being added at each time step. So we're just redefining a Markov matrix so that we can taper how frequently those mutations are occurring. Okay, great. So now we have this iterative way of going from x naught to x at small t. And now we want that second criteria of measuring a sample at any time t given x naught. And again, we can envision how this links to a Markov process where we take that sample at time 0, we multiply it by this cumulative product of Markov matrices, and then we get our sample at time t. Okay, great. And now we have, again, this defined way of sampling x of t, given only x of 0 and our scheduled transition matrix. Now, yeah, go.
Could you go back a couple slides? I just wanted to make sure I understood this. So it looks like you're doing a transition where with some probability, you stay where you are, and with the complementary probability, you just completely, it's just completely random?
Yes, yeah.
So, okay.
Yeah.
Right,
and yeah, and I think I'll touch on this in a little bit, but that choice of the probability at which you transition to something else is up to you as a user. So that is something you can control.
The beta, so does that become a tuning parameter eventually?
It's a model choice. It's a choice you would make about your corruption.
Okay.
And Ava, I think, will show a nice example of that later, but I'll also touch on that in a second.
Thanks.
Okay, and so now we can go back to this simplified training objective, and what we find is that a lot of these components of this function are the same in discrete case. So for example, at time t, we're again looking at how close this is to a uniform sample. We can ignore this during training. We have this reconstruction term that we know how to handle, and now all we have to do is define a way of describing this term here. And this was done in a paper in 2021 by Jacob Austin, where they derived this, the terms in this KL divergence, which is a comparison of two distributions, using these various Markov properties that depend on the input sequence and the Markov transition matrix, which in this case is represented as big Q, but I was calling M earlier. And the key thing here is that we have now these three terms that represent this objective here. They also add in this weighted cross-entropy loss, giving our final loss function to take on this form, where we take our lower variational bound term, plus some weighted version of our cross-entropy. This is just how well our model reconstructs our inputs. And we can control that by altering this lambda term here. So this is just a constant from 0 to 1. Okay, great. And now we have another loss function that we can train on in the discrete case. So again, we're going to go through this simple training procedure in which we can train our model. So again, we pre-compute those terms that describe our variance schedule. We're going to randomly sample some time t, and we're going to generate our prediction at that time t. We do the same kind of training procedure again, where now we have our discrete loss function, and we're, in this case, because of the way that the loss function is described, we're predicting our inputs directly at each step instead of predicting the noise being added. And that's just built into the way that that Austin's defined this loss function. And again, we just repeat this until our model converges. Again, we have kind of the same process here of a generative sampling procedure where we start with some random sequence, pass it through a model, and iterate inverse the samples at inverse time steps. Again, we need a little bit of math for this. We have defined this previously in our loss function, but we're able to do this iteratively until we get a new protein sequence. And now that we've discussed how we can train a continuous diffusion model and a discrete diffusion model, we can think about some of the considerations we need to think about when we're designing proteins. Now, we have considerable public data sets of protein sequence, structure, and function that fit into both of these frameworks. So for our continuous models, we can imagine the fact that we have hundreds of 200,000 crystal structures available on pdb.org, and we could model these 3D representations of proteins in a continuous fashion. Similarly, in the discrete case, I've given this example on protein sequences. On UniProt, there's around 250 million proteins. UniRef50 is the subset of non-redundant sequences in this data set. And what we can see here, and a lot of these are annotated as well, which fit nicely into this discrete framework of having a textual representation of the function as well as the kind of a textual representation of the protein sequence as well. But there's some things that we need to consider when we design both of these. When we think about continuous protein diffusion, there's one criteria that we need to ensure is true, and that's that our model is invariant to rotation and translation. Meaning that if I translate my protein in XYZ space, or if I rotate it, we know that these 3 sets of coordinates all represent the same protein, but we need to ensure that our model does as well. There's a few different ways of doing this. Typically, we can represent proteins as these 3D point clouds where we take their heavy atoms, their alpha carbons, and we represent them as their XYZ coordinates. These are obviously invariant to These are obviously will fall victim to translation or rotation, as those digits will change. But we can represent our proteins in, say, a different description, for example, using angles or dihedrals. And this now is invariant to those transformations because the value of those angles or dihedrals don't change if we move that protein in 3D space. We've done some work, well, I haven't done work on this. Ava has done some work on this in a paper that came out recently called Folding Diff, and she'll show you a little bit about this later today. But say you don't choose to do this new representation of angles and dihedrals, you then need to build in these considerations into the way that you design your models. So one example of doing this is using something called an SE3 transformer. It uses the self-attention mechanism that's in transformers to ensure that your inputs and outputs stay invariant to these transforms or rotations. There's 2 major examples of doing this. One is RF diffusion. It's this new state-of-the-art protein generative model that came out of the Baker Group. And then Namrata Anand published something in 2022 with Tudor Akim that used this same mechanism to account for equivariance. Now, some considerations in the discrete protein diffusion case are in how we might represent the data. So I think everyone's kind of picked up on this idea that we can control the mutation, and a random uniform transition matrix is not the most intuitive way to transform proteins. So can we change the way that we mutate our proteins by defining a new transition matrix? Or rather, can we even replace that with something like an absorbing state that iteratively mass out residues until we're at a fully masked sequence, and then generate proteins in that way. We published a paper on this on bioRxiv just a few months ago, showing this exactly. And again, Ava will present this in a little bit. The next thing you might think about is your model choice. So what types of models you want to use to train your data on. And the two major ways of doing this are with transformers or convolutional neural networks. We find that convolutions or CNNs can handle these longer sequences a lot more efficiently in terms of memory and compute. And as a con, and something you have to think about if you want to implement transformers, is that they're position invariant. So meaning you need to provide some additional encoding telling the model at what residue each amino acid is at in the sequence. And Kevin Yang, who's in the audience, has a nice paper outlining kind of the trade-offs between the two here, showing that convolutions are actually a fair model to use in this case. And then lastly, we have, we're starting to enter this era where we want to combine data from both sources of both structure and sequence. This notable paper came out recently where it does sequence diffusion conditioned on structures. But you can imagine that we might also be interested on diffusing in both sequence and structure space at the same time. And that would be a natural next step here.
Today, I'm going to talk more specifically about our efforts in our research in leveraging these generative diffusion models to optimize protein design with respect to both structure and sequence. So before I do that, I think what brings us all here today is a common interest in this idea of models, inferences, and algorithms, right? Bringing computational and statistical tools to bear on the largest problems in biology and medicine. But I want to start by taking a step back, or rather a dive deep down, to think about really what is it that we're considering here, and to remind you all of the complexity and richness of biology at the molecular level up across scales of resolution. So what I'm showing you here is a visualization from this wonderful book, The Machinery of Life, by scientist and illustrator David Goodsell. And it's an artistic rendition of a 1 million times magnification of the nuclear membrane and the nucleus in the cell, and all the protein machinery that is operating at this level. And so, right, as computer scientists, mathematicians, statisticians, we want to bring our toolkit to these problems in biology. But there's this incredible complexity that we're faced with, and we're trying to understand these systems really from the bottom up. And I think at its core, this idea of generative modeling gets to the very heart of a scientific question, where I think that the physicist Richard Feynman put it best, where he said, what I cannot create, I cannot understand. And really, that's kind of our overarching vision with this new era of generative design and generative AI, where our hope is that by leveraging these tools, maybe we can discover something fundamental about the biology of these systems, and also design new entities that will perform useful functions for us as humans. And really, that's our unifying goal in our team at Microsoft Research, where our mission is to develop new AI systems that can actually contribute to new biomedical knowledge through this lens of generative design and interactive discovery. And we're considering this in our team across scales and resolutions of biology, all the way down from the molecular level, where we'll spend our time today, up to the patient level, and thinking about higher level questions at the context of the individual patient. And so today, as Sarah introduced, and as we'll dive in, we're going to really focus on questions in protein engineering and how we've developed new modeling frameworks for designing protein structures and sequences. So why do we want to design new proteins? Why do we care? The fundamental motivation here is that we can expand the scope of functional space that's occupied by proteins, which are the fundamental actuators of biology, by hopefully engineering and designing them to do new and useful things. Now, there are already several key examples of this in our world today, from molecular tools like GFP, to enzymes that can actually catalyze new chemistry that doesn't occur in nature, like this enzyme cytochrome C, which can catalyze the formation of carbon-silicon bonds, and ultimately to design new therapeutic modalities that could hopefully eventually be deployed in the clinic, like this example, which is an enzyme that's used actively as a chemotherapy in leukemia. And so across all these examples, the fundamental question is, how can we design novel proteins to expand our capabilities functionally with biological, therapeutic, or other applications? And the goal of generative models and learned neural network models that aim to try to accelerate this task is to look at data and examples from real biology and learn from this diversity and learn from these data sets to then ideally generate designs of new proteins that can perform these new functions. And as I kind of introduced, our goal is not only to go forward from biology to a generative model and newly designed proteins, but hopefully to try to leverage this as a framework by which we can better understand the biology of the systems themselves that we've been considering. And so today we're going to dive into some of our recent work in our team, where we've developed these diffusion models that are biologically grounded, whether in the biophysics of protein structure or in the evolutionary landscape of protein sequence, to try to optimize and guide protein design across these levels. And I'll draw your attention to this kind of canon of the dogma of protein function, where we know that proteins are fundamentally encoded by sequences of these amino acid building blocks, where we can think of this as a language. And that sequence then dictates an overall three-dimensional structure to the protein that then dictates its eventual function. And so first, I'm going to focus and introduce a little bit about our work in developing diffusion models of protein structure. And so here, we really wanted to ensure that the model that we developed was grounded fundamentally in what we know about the biology and the biophysics of protein structures and how they are realized. And so for this, we drew inspiration from how proteins naturally adopt their final 3D conformations and structures in the cell, which is the biophysics of the protein folding process. And so at a high level, right, after a protein is translated as it's being made, it can adopt a kind of loose, random, unfolded configuration. And during that folding process, the chain of amino acids will twist and sample different conformations to finally arrive at a final folded 3D configuration that's energetically and thermodynamically stable. Now, the bonds that connect the backbone atoms in this protein structure are actually defined geometrically in a very specific way. In fact, we can precisely describe the angles that exist between these individual atoms that exist in the protein and use this set of bond angles to completely specify and delineate the overall structure of the protein. And so taking a step back from this point of view of how these angles between atoms in the protein backbone define a structure, we can kind of simplify this and say, okay, well, at an unfolded state, let's say the angles in this protein structure adopt more or less a random configuration. But we know that at a final folded structure, these angles are highly structured that actually confer the protein's overall 3D geometry and its structure. And so looking at these settings side by side, where we have something noisy, random, and something structured and folded, we asked the question, can we learn a generative model that can allow us to translate between these two? And this was work that was really creatively spearheaded and inspired by a former intern in our group, Kevin Wu, a former graduate student at Stanford, who raised this question of, what if we think about protein structure as this configuration of 3D angles? Can we learn a generative diffusion model that can operate by going from a completely random set of angles that specify an unfolded state and learn to denoise from this to then generate new sets of bond angles that would ultimately specify a 3D folded structure. And indeed, you can see a visualization of that iterative denoising, that iterative generation process occurring here, where we have started with a random configuration, and over this iterative denoising process that constitutes the diffusion model's backbone, we can arrive at sampling this final structured configuration. And so this is the essence of this structure-based diffusion model that we developed that we call folding diff, short for folding diffusion. And the principle is very much in line with the framework that Sarah introduced in the primer, where now we have this process of taking real data, in our case, the sets of bond angles that specify the geometry of the protein backbone, iteratively noising them in the forward process to arrive at a random set of angles corresponding intuitively to an unfolded state, and leveraging these iterative noised examples as the way we can train a generative diffusion model to now denoise, and ultimately learn to reverse this process to then be able to sample new sets of bond angles that will generate realistic structures. And what is key to this representation and this formulation is that because of the fact that we're operating over the bond angles that specify the geometry of the protein backbone, all the nuances about the symmetries and invariants under translation and rotation that Sarah introduced are fundamentally encoded in this angle-based representation itself. And so we can use a simple neural network architecture that doesn't have any of those constraints baked in, just like a vanilla transformer, to actually constitute that neural network model that learns this denoising process. So to make that a little bit more intuitive and visual, in this example here, with our formulation where we're operating over these internal angle coordinates, the folding diffusion approach takes that equivariance requirements and encodes it within the angle-based representation itself. And this is in contrast and complementary to other approaches where now treat the structure as not a set of angle coordinates or angle sets, but rather as points in 3D space that are specified by 3D coordinates, where then you have to encode the equivariance requirements into the neural network architecture itself. And so these are two complementary approaches in terms of how we can think about formulating structures of proteins. And there are other representations that have also emerged recently. And so I'll take you through a little bit of our empirical results that we show with our folding diffusion approach. And we primarily considered, okay, now that we have this formulation, can we effectively generate sets of bond angles that recapitulate the distribution of natural protein angles? Can we generate structures that preserve key structural motifs and are qualitatively good? And furthermore, are these structures actually realizable with an amino acid sequence? And are they diverse with respect to the structures that they adopt? So I'll give you a little bit of taste of that, of some of those results, where first we considered the ability of our modeling approach to actually capture the natural distributions of the angles we see in known proteins. And indeed, these bond angles are not completely independent, right? They're highly correlated in their distributions that eventually specify key structural motifs. So if we compare the distributions of the two dihedral angles, phi and psi, of true protein backbones versus those that are generated from our folding diffusion model, we see that we are very well able to capture the patterns and correlations across these backbone angles, indicating that our method can capture and learn patterns of these key structural motifs entirely in this angle-based space. And indeed, this is recapitulated in the quality of the structures that we're able to generate from folding diff, where we can see qualitatively we are able to capture overall geometries, structural motifs like alpha helices and beta sheets, and that these structures are notably distinct but yet similar to similar structures from the set of natural proteins. We get into more quantitative evaluations in the paper as well, where we assess the quality of these structures in terms of their structural diversity, how well they can be physically realized by amino acid sequence that will specify a given fold. But for the sake of time and for focusing on some of our more recent work, I would like to refer you all back to our paper, as well as our code base and open source models, where you can play around with the folding diff approach and sample new generations.
Hey, Ava,
yeah.
This is really cool. Before you move on, have you ever fed through your generative model proteins for which we know the folding pathway, or at least no intermediate states along the pathway? I'm curious if you've done that, or you think the model would kind of pick up those sort of intermediate folded states, or is it going through some kind of other pathway of folding?
Yeah, that's a great question. The question was about whether we can specify intermediate states and generate from there.
But actually, that's also a good question. But I was more just like, even without generating, just if you took a sequence where you know how it folds and just look what you see on the way, does it look like what we see experimentally?
Okay. Yeah, so in this case, all the generations are completely unconditional. So we don't specify any prior about what we think the sequence or the structure should look like. That indeed is something that we are interested to investigate further. I do know of a couple other groups who have started with our folding diff approach and have specified sets of protein candidates that they're interested in, and have used this framework to now basically fine tune on those sets and show that they can generate things that are similar to the data they have at hand. Awesome. So this is entirely right in structure-based space with this angle-based formulation. And I've told you a little bit about why we care about structure, and fundamentally the generation of backbones that can specify particular geometries that we care about. But the protein design problem spans these different scales of resolution that we can think about, right? We can think about starting with the language of amino acid sequences and what is encoded there, all the way up to hopefully being able to think about specifying particular functions and then designing according to functional constraints. And so we were really motivated by this paradigm and asked, okay, well, structure takes us so far, but what if we could actually leverage the foundational language of protein biology in amino acid sequence space to now be able to dictate controllable design with these diffusion modeling frameworks? And our reasoning and our rationale for that was the fact that we hypothesized and reasoned that sequence could actually serve as a more universal design space for protein design. In structural design, as I introduced before, we can specify a geometry of the structural fold that we want the protein to adopt. But ultimately, in order to realize that geometry, we require an amino acid sequence to actually synthesize and encode that protein that hopefully specifies that structure and geometry. And so there's this indirect step of requiring sequence design whenever you design a new structure. Secondly, a lot of our current bioinformatics tools in terms of how well we're able to predict the likely function of a protein are built off sequence space, meaning that these strong function predictors, whether they be classifiers or regressors, are typically trained over a much larger scale sequence data sets that are then paired with functional annotations. And really, that gets at the heart of the fact that this is fundamentally a problem of data and coverage as well. in that we only have solved crystal structures for approximately 200,000 proteins in the PDB, whereas we have much, much larger data sets of unannotated protein sequences that cover a much greater diversity of evolutionary space. And finally, and also critically, there are regions of the functional space that proteins occupy that don't fit nicely into the traditional structure-based paradigm. Importantly, a lot of important biology in the cell is encoded and enacted by these intrinsically disordered regions in proteins, which are experimentally very difficult, right, to resolve structures for, and therefore not easily accessible by structure-based generative models. And so with these motivations, we reasoned and asked, could we eliminate structure entirely as a variable in this generative modeling framework, and instead reason entirely over sequence space by building diffusion models of over protein sequence space to achieve controllable design. And this is work that was a very large team effort from our team at Microsoft, but really spearheaded and led by Sarah, who really led the hands-on work and the technical execution for this project. And so we built a discrete diffusion modeling framework that enables us to generate over amino acid sequence space for protein design. And we call our method EvoDiff, which stands for evolutionary scale diffusion. And critically, what we do with EvoDiff is we consider now not structures, but sequences of protein amino acids. And we develop different noising and corruption screens to be able to learn these generative diffusion models to now sample new generations. And specifically, connecting back to the framework Sarah introduced in the primer, we explored a couple of ways to actually do this in this discrete setup. The first is called order agnostic autoregressive diffusion, or OADM, where we start with an amino acid sequence, and then stepwise, we corrupt individual amino acids in this sequence by the form of a masking token, a special mask token. And then use these examples to train a neural network denoiser, where then we can learn to predict the unmasking of these individual residues to then ultimately uncorrupt and denoise from a masked state. And so to provide you a visual of how this works, you can think about, we start with an amino acid sequence, we select randomly in a completely order agnostic fashion an individual residue to mask, we do this stepwise, and the network is learning to predict the identities of those amino acids at those mask positions. Importantly, this masking can occur in any arbitrary order and is agnostic to the actual order of these mask tokens, such that then at inference, when we want to sample new generations, we can start from a sequence of all mask tokens and be able to decode stepwise starting from that position. We also explored a second scheme for this discrete diffusion framework that is called D3PM, where now, instead of introducing a specific masking token, we are mutating the identities of individual amino acid residues according to some defined corruption scheme. And there were a couple of questions earlier in the primer about how we actually choose this mutation scheme. The first and most straightforward way is to say, okay, with any amino acid can change to any other amino acid with equal probability, meaning a uniform way to corrupt these sequences. Or we can try to be a little bit more clever and select a corruption matrix that is based on known frequencies of amino acid substitutions that are found in nature. And interestingly enough, while we thought that this bioinformed scheme, this blossom-based scheme, would lend a substantial difference in performance, in practice, empirically, we found that there was not a significant difference between the two schemes. And I think that boils down to the fact that it is actually a pretty hard task for these models to learn. And the reason for that is, looking at our visual here, when these individual amino acids are mutated, the network doesn't have an explicit signal of which positions were changed. Unlike the masking scheme where you have that specific mask token, the amino acid mutations are just changing, they're just mutating. So it's much harder for the network to actually learn what that residual corruption is. And at least this is our hypothesis about why this framework is relatively a harder task. And so I won't get into the exact benchmarks in comparing these two schemes, but as I kind of alluded to, we found that empirically the masked order agnostic corruption scheme performed much better than this mutation-based scheme. And so we call our models here EvoDiff-seq, using this mask corruption framework and denoising framework. And this will constitute the base model for all the empirical results that I show. So if you have, oh yeah, question.
If you're always starting sort of with like a fully masked sequence, then like how do you generate different amino acid sequences?
Yeah, so the question was, if you start from fully masked, how do you generate different diversity? And it relates back to this notion of, what is the stochasticity and what is the randomness in these iterative steps, right? Remember that the denoising is not deterministic, right? The network has just learned a distribution over this denoising function, right? So stepwise, there's going to be differences in what is chosen to be uncorrupted at each time. And therefore, you're going to get variability step by step. And so unconditionally starting from all masked, you can generate diversity. And you can also mask portions of the sequence to start and guide the generation in this way, which I'll also touch on.
When you tokenize amino acids, can you what if you added a dimension to indicate which amino acids are mutated, and then the network would have some signal for which amino acids to,
Yeah, that's quite an interesting suggestion. To indicate, have some encoding about the position. We did not experiment with this. I think the rationale was that in this corruption scheme, we ideally don't want to specify that to allow for basically as much capacity, which also creates a more challenging learning task, but that could be interesting to play with to constrain one. Yeah.
When you since you start with everything masked, how do you decide which amino acid or which residue to flip first?
The question is which residue is chosen to flip first. This is a random choice. It's a stochastic in the denoising process, right? Which is the virtue of this completely order agnostic scheme, because of the fact that the model has learned any possible decoding order and any possible combination of masked positions.
Yeah, I understand. But is it like uniformly random at the position level, or you actually have a learned distribution over the residues, like based on their learned logits, for example?
I see. Yeah. So initially, I believe we started with completely uniform at random, but also we did, I think, some we can decode based on the entropic consideration, right, of what positions carry more information and preferentially decode according to that. So we could specify the order. Yeah.
I have a question. My name is Lucas. I'm curious about the way you encode the amino acids. We know the amino acids have some relationship, like chemical relationships, and how they can interact in the different proteins. So if they are in the substructure, like like alpha, like you're gonna have a combination of amino acids that always gonna be there. So I'm curious, could you, instead to encode just like a tokens, like without any meaning or chemical meaning, like put this in the embedding or something like a model that could already tell you about this relationship? Do you think this would improve the model with speed? Yeah, that's my question.
Yeah. So if I understand correctly, the question is about how are we capturing information about the chemical properties of these individual amino acids, if at all.
Yeah.
This is the virtue of training on very, very large sequence data sets, where the hypothesis is that information about the properties of those amino acids and their relationships to one another is going to be latent in these large, very large sequence data sets. And so there is no explicit encoding of chemical identity beyond just encoding the individual identity of the individual amino acids. That is to say, we don't say, oh, this is a hydrophobic amino acid, this is a hydrophilic amino acid. In this case, we're learning purely from sequence data in a completely data-driven manner.
Thank you.
Okay, so I'll briefly, maybe I'll, I think that transitions well to this point, where now that we have this diffusion framework, how do we act, what is the data set we actually leverage to learn this model, and why is that important? The key component of EvoDiff beyond this diffusion modeling framework is the fact that we train it on an evolutionary scale set of sequence space. Specifically, we have a data set of 50 million unique protein amino acid sequences that are taken from proteins with functions and sourced from organisms across the tree of life. And so that's really a key aspect of our modeling approach, where we reasoned that by leveraging this diversity, we could learn effective representations that would capture diversity in function. And the backbone neural network we use to learn this denoising operation is a convolution-based architecture to perform the actual learning. And so to give you a little more intuition behind how this generative process actually works, we can start from all mask tokens, and then at inference time, leverage the trained EvoDiff model to do this iterative denoising. And what I'm visualizing on the left is entirely the denoising operation of EvoDiff purely in sequence space. And on the right is the predicted structure resulting from that sequence. And so importantly, our model is only looking at sequence. The visualized structure is purely for the sake of visualization. And so to get a little more into the details about our empirical results with this framework where we're combining this evolutionary scale training with discrete diffusion, we wanted to consider how good our generations were, primarily along 3 axes. First, are these proteins structurally and synthesizably plausible, meaning would they adopt stable structures and fold into those stable structures? Second, how diverse are they? How novel are these sequences? How well do they cover the spectrum of protein functional and structural space? And finally, can we perform different forms of conditioning to now try to control the design of these proteins according to desired constraints? And so the first thing that we wanted to consider was how realistic and plausible are these structures? And to do that, we took our trained EvoDiff model, sampled from it to generate 1,000 different and unique sequences, and then passed each of those sequences into a structure prediction tool like OmegaFold or AlphaFold, and evaluated the confidence in the stability of that predicted structure. We also performed an additional step where from that structure, we can predict an inverse folded sequence likely to adopt that structure, and then compare, cross-match our original generation with that prediction to see how consistent these are. And so what we're visualizing here on the x-axis is the confidence in the prediction of that structure against the consistency of those two sequences. And our premise here is that by considering these two metrics, we can assess how plausible these proteins are. And so first, visualizing examples from the independent test set of natural proteins, we can see that there is a spectrum of structural confidence and sequence consistency that generally correlate with each other, and that generations from EvoDiff occupy this distribution pretty well. Importantly, that we get a good number of generations that are both predicted to be high structural confidence and self-consistent. We can also inspect these generations qualitatively across different amino acid lengths, where we can see from a few selected examples across lengths that these proteins are structurally realistic, that they have high confidence and low similarity, meaning that they are diverse from known sequences and structures of natural proteins. Importantly, when we compare our generative diffusion framework that operates over sequence space to other approaches for protein sequence design, we can see that we perform well, if not better, to compared state-of-the-art approaches. Namely, in comparing traditional left-to-right autoregression, where we decode in a fixed order going from a start amino acid all the way to the end, we can see that we perform competitively with respect to this plausibility. And secondly, compared to masked language models of proteins, like a model like ESM2, we can see in general we perform better in terms of generating sequences that are plausible. And importantly, this can be attributed to the fact that our EvoDiff model, as well as the traditional left-to-right autoregression, are both trained explicitly for a generative task, whereas masked language models are traditionally not. So that gets a little bit at a flavor of the sense of plausibility of structure and sequence. The next thing we wanted to evaluate is how well the coverage we had over functional and structural space and how diverse these generations were. So what we did to assess this was we took sequences from the natural set of proteins, our test set, and used a tool to generate embeddings that have been shown to capture functional properties and recapitulate differences in protein function. And we can visualize that in 2D space using something like a UMAP. We can do a similar approach for generations from EvoDiff, and we see that overall we have relatively good coverage over this representation of functional space. But as you'll note, there's this really interesting part of the space that's completely missed by our generations. And this is something that we're seeking to follow up on. We can also compute quantitatively how similar these embeddings are and come up with a distance-based metrics that captures that. And these metrics are useful, but only useful if we contextualize them with respect to other baselines and other tools out there. And so doing that, we see that EvoDiff performs competitively with respect to left-to-right autoregression, although traditional autoregressive models provide a better coverage of this functional space. But note that our diffusion framework will enable us to do conditional generation, whereas the fixed decoding order of left-to-right autoregression, or fixed generation order, excuse me, of left-to-right autoregression does not. Compared to a masked language model, we perform substantially better, achieving higher quality generations. And compared to the state-of-the-art structure-based generative model, RF diffusion, we achieve greater coverage of this functional space, attributed to the fact that we're learning from a much greater coverage of protein data at evolutionary scale. So this gives us a sense of function. We can perform similar analyses where we generate embeddings and evaluate these distributions for structural features using another structure-based another predictive tool that will generate embeddings representative of secondary structure elements and annotate individual residues for predicted secondary structures. And we see here that EvoDiff, again, does a good job of capturing the known distribution of secondary structure elements better than tools like masked language models or structure-based diffusion. Okay, so in the remaining 15 minutes or so, I want to focus the remainder of the talk on some of our first attempts at doing conditional design and conditional generation using our EvoDiff framework. And specifically, we leverage the fact that we are doing this order agnostic decoding and corruption to our advantage. Because of the fact that we can mask and then decode in an arbitrary order, we can do conditional generation by fixing some parts of a protein sequence and then asking the model to look at the masked remainder and then in-paint or denoise over that remainder. And so what this allows us to achieve is basically a form of in-painting where we can specify the particular residues that we want to be masked and decoded and ask the model to generate new samples over those positions. And so this can take a variety of forms. And we explored several, and we'll touch on a couple in the talk. The first we asked was, can we leverage explicit evolutionary information about proteins from the same family? Where now we specify not only a single sequence to the model, but rather an alignment of multiple sequences of evolutionary related proteins. And by simply masking the reference sequence or the query, we can then ask the model to generate over this masked query portion and hopefully generate a new protein that will be structurally similar to those other homologs specified in the alignment. We can also then leverage masking of particular portions of single sequences or of alignments to now perform in-painting or scaffolding, where we can operate entirely in sequence space to then condition the design based on these portions that we want to hold fixed and the portions that we want to keep variable. So I'll give you a taste of some of our results in this framework that we leverage multiple sequence alignments of proteins to do this more explicit evolution-guided generation. The concept here is where we take a protein sequence of interest and we can construct a multiple sequence alignment or an MSA by searching for proteins that are related to that sequence and subsample down to a reasonable set that our model can work with. And then leveraging that same concept of this masked discrete framework, we can now operate over the entirety of the MSA, not just a single sequence, by performing the same scheme of this iterative corruption and then training a neural network model we call EvoDiff MSA to learn this denoising operation now over these multiple sequence alignments. And the way that we can do that is we can leverage already existing great backbone architectures that operate over these multiple sequence alignments, specifically tools like MSA Transformer, to now constitute the backbone for our neural network denoising. What is so powerful about this approach is that this EvoDiff MSA, by leveraging this evolutionary scale information and this explicit information in these alignments, can function as an amortized local generative model. So to explain what I mean by this, if we want to generate sequences related to a protein family that we care about, the standard approach would be to take sequences from that family and train a bespoke generative model on that data and then ask it to generate new instances. Instead, with EvoDiff MSA, because it's leveraged all this evolutionary information, we don't need to train a bespoke local model. we can just now specify that multiple sequence alignment of related proteins, mask the reference query, and now use this at inference time to produce a generated sample that will hopefully be related to that information we specified in the alignment. And we can see that this process actually improves the quality of our generations in terms of the plausibility, again, looking at the structural similarity and sequence self-consistency as before. And importantly, we can compare how well those generations capture the original structural fold specified by that related family. So are we actually conserving the structure that was specified in our local alignment? And we find that, yes, indeed, with EvoDiff MSA, we're able to perform well in terms of capturing that specified structure that's faithful to the original reference sequence, while still generating new sequences that are novel and distinct from the information that was found in the multiple sequence alignment. And so, again, we can perform evaluations both qualitatively and quantitatively to see the quality of these generations. So in the final portion of the talk, I will share a little bit about what I think are perhaps the most exciting results from this work, in that supplying a different but related form of conditioning, where now, related to some of the questions that we've been discussing, we're fixing particular portions of the sequence in any arbitrary order and asking the model to now generate diverse sequences across the remainder. And this is motivated by the fact that a lot of protein interactions in biology are mediated by a notion of a functional motif that we usually think of as occupying a particular structure that interacts with a partner, and that functional motif is somehow stabilized in the overall scaffold or the overall structure of the protein. And traditionally, in our current thinking in the field, the majority of approaches have taken a structure-first approach to tackling this question of designing new diverse scaffolds that capture a functional motif. But we ask the question, because of the fact that we leverage this evolutionary scale information, can we leverage this data to actually scaffold functional structural motifs without specifying any explicit structural information? Meaning, can we do it from sequence alone? And the answer to that, perhaps because I'm posing it, is yes, resoundingly yes. And so what we can do is leverage our discrete diffusion framework to consider a protein example, where what I'm highlighting here in green is a functional motif that specifies a binding site of this protein. And we can keep that portion fixed and then take out all the other residues of the native protein. And now keep that functional motif as conditioning information, mask the remainder of the sequence, and ask EvoDiff to now generate the remainder in the same framework that we described previously. And excitingly, we can find that we do this very well. We can generate new diverse sequences that occupy the same overall structure, that preserve the structural information present of that motif. And we do this entirely in sequence space without any explicit structural information. These are a couple of select examples with generations of this functional motif scaffolding from both our sequence model and our MSA-based model. And excitingly, as what you can see, right, these motifs are not continuous in sequence space. They're not continuous in structural space. And yet we can generate novel sequences that are distinct, but high confidence in their structure, and also preserve the position of that motif with integrity. We evaluated over a set of benchmark problems that were initially proposed by a structure-based generative model, RF diffusion, And what we can find is that we perform very competitively to this state-of-the-art structure-only model, leveraging our approach in terms of the number of these scaffolding tasks we actually generate viable structures for, and how well they preserve those key motifs. And so we can happy to talk more about the details of these empirical results and some of our quantitative evaluations here. But for now, I just want to close and summarize with the fact that with this diffusion, combined the power of these evolutionary scale sequence data sets with this diffusion modeling framework to enable both high fidelity, unconditional generation of new protein sequences that are structurally plausible and diverse, demonstrated that we can do conditional design with scaffolding of functional motifs entirely in sequence space. And moving forward, we're quite excited about leveraging this framework to now explore different forms of conditioning to ultimately be able to get at the point where we can specify desired functions and ultimately guide the design of new proteins according to those constraints. So with that, I really want to emphasize that these efforts that we shared with you today are just one part of our broader effort in biomedical machine learning at Microsoft Research. And ultimately, our goal is to leverage these models and frameworks that we develop all in conjunction and deploy them for high impact biological and medical problems, and always open to think about potential collaborations and ways that we can achieve that.